In the year 1492, when Christopher Columbus first stepped onto the soil of what he mistakenly believed to be the Indies, he carried with him not merely the ambitions of Spanish monarchs or the weight of navigational instruments, but an entire paradigm of understanding the natural world that would shatter upon contact with the botanical realities of the New World. The sailors who descended from those 3 ships into the warm Caribbean waters had never seen a pineapple, had never tasted chocolate, had never witnessed the spectacle of a ceiba tree rising 200 feet into the tropical canopy, its massive buttress roots spreading like cathedral foundations across the forest floor. What they did not know, what they could not have known as they gazed upon these strange and magnificent plants, was that they stood at the threshold of a revolution that would transform not merely cuisine or medicine, but the very structure of global power, the patterns of human migration, the rise and fall of empires, and ultimately the composition of breathable air across the entire planet. The indigenous Taino people who encountered these bewildered Europeans possessed knowledge of plant cultivation that had been refined across millennia, an understanding so sophisticated that it would take European botanists 3 centuries to even begin to comprehend its complexity. They grew cassava, a plant whose roots contained enough cyanide to kill a man outright, yet through an intricate process of grating, pressing, and heating, they had learned to transform this toxic tuber into a staple food that could sustain entire populations. The sailors from Seville and Palos could not have imagined that this very plant, manioc as they would come to call it, would one day feed more than 500 million people across Africa, South America, and Asia, becoming the 3rd largest source of carbohydrates in the human diet behind only rice and maize. Columbus himself wrote in his log of the roots-like carrots that the natives ate, a description so inadequate it barely registered the magnitude of what he had encountered, like describing the ocean as rather wet. But before Columbus, before the age of sail and gunpowder transformed the world into a single, interconnected marketplace of botanical exchange, plants had already been reshaping human civilization in ways both subtle and catastrophic. In the 13th century, as Genghis Khan's mounted archers swept across the steppes of Central Asia, the grass beneath their horses' hooves was performing a conquest of its own. The grasses of the Eurasian steppe, feathergrass, fescue, and wild wheat, had evolved over millions of years to withstand the thundering passage of massive herbivores, their roots forming dense mats that could survive the most intensive grazing, their seeds adapted to dispersal in the digestive tracts of animals, or caught in their fur. These grasses made possible the great horse cultures of the steppes, providing the fodder that sustained the enormous herds necessary for nomadic warfare. The Mongols understood this relationship with an intimacy that sedentary peoples could never quite grasp. They knew which pastures could support 1,000 horses for a week, which plants indicated water beneath the soil, which grasses would fatten their mounts most quickly before battle. This botanical knowledge was as crucial to their military success as their famous composite bows or their revolutionary system of military organization. Meanwhile, in the mountains of Ethiopia, a shepherd whose name has been lost to history noticed that his goats became unusually energetic after eating the bright red berries of a particular shrub. This observation, probably made sometime in the 9th century, would eventually lead to the global coffee trade. But the path from that Ethiopian hillside to the cafes of Vienna and Paris was neither direct nor simple. The plant, Coffea arabica, grew wild in the cloud forests of the Ethiopian highlands, thriving in the cool, misty conditions between 5,000 and 7,000 feet elevation. For centuries, the knowledge of coffee remained largely confined to the Horn of Africa and southern Arabia, where Sufi monks discovered that a beverage made from the roasted seeds could help them stay awake during nighttime prayers. The Yemeni port of Mocha became the sole gateway through which coffee beans entered global trade, and the Ottoman Empire guarded this monopoly with obsessive care, going so far as to roast or boil all exported beans to ensure they could not be planted elsewhere. In 1616, a Dutch merchant named Peter van den Broeke managed to smuggle living coffee plants out of Yemen, breaking the centuries-old monopoly and setting in motion a chain of events that would transform Java, Ceylon, the Caribbean, and eventually Brazil into coffee-producing regions. But the real revolution occurred not in these new plantations, but in the European cities where coffee houses became centres of radical thought, commercial innovation, and political conspiracy. The English coffee houses of the 17th century were called penny universities, because for the price of a penny, the cost of a cup of coffee, a man could listen to extraordinary conversations, read newspapers and pamphlets, conduct business transactions, and encounter ideas that would have been dangerous to discuss in more traditional settings. In these establishments, merchants created the foundations of modern insurance at Lloyd's Coffee House, scientists demonstrated experiments and debated natural philosophy, and political radicals plotted against the crown. King Charles II attempted to suppress the coffee houses in 1675, claiming they were hotbeds of sedition, but the economic and social forces they represented had become too powerful to contain. The very physiology of coffee consumption, the way caffeine stimulates the central nervous system, increasing alertness and concentration while reducing the perception of fatigue, was reshaping human consciousness and productivity. Unlike alcohol, which had been the primary social beverage of medieval Europe, coffee allowed people to gather and converse while remaining sharp and clear-headed. Some historians have argued that the Enlightenment itself was partially fueled by the transition from alcohol to coffee as the beverage of intellectual discourse, a pharmacological shift that enabled new forms of rational thought and systematic inquiry. Yet while coffee was altering the mental landscape of Europe, another plant from the Americas was engineering an even more profound transformation of global economics and human suffering. Saccharum officinarum, sugarcane, had originated in New Guinea and had been cultivated in India for thousands of years. But it was the European colonization of the Caribbean and Brazil that transformed sugar from a rare luxury into a commodity that would reshape the world economy and create one of history's greatest atrocities. The process of growing and refining sugar was extraordinarily labour-intensive. The cane had to be planted by hand, required constant weeding and irrigation, and once mature, had to be harvested quickly with machetes before its sugar content declined. After cutting, the cane had to be crushed immediately to extract its juice, which then had to be boiled in a precise sequence of copper kettles to concentrate the sugar, a process that required skill, timing, and brutal, unrelenting work in conditions of almost unbearable heat. The profits, however, were staggering. In the 18th century, the small Caribbean island of Haiti, then known as Saint-Domingue, produced more wealth than all 13 American colonies combined, its productivity built entirely on the cultivation of sugarcane by enslaved Africans, who died at such appalling rates that the island required a constant influx of newly captured people to maintain its workforce. The death rate on sugar plantations was so extreme that in most Caribbean colonies, the enslaved population could not sustain itself through reproduction. Only the continuous importation of human beings stolen from Africa kept the system functioning. This was not an accident or an unfortunate side effect, but a mathematical calculation made by plantation owners who determined it was more profitable to literally work people to death and replace them than to maintain conditions that might allow for survival and family formation. The sugar that sweetened tea in London drawing rooms and made possible the elaborately decorated cakes of Parisian salons was stained with a horror that most consumers never witnessed or imagined. The plant itself, Saccharum officinarum, was simply a large grass that had evolved in the tropics to store massive amounts of sucrose in its stems as an energy reserve, a botanical adaptation to seasonal climate variations. It had no moral dimension, no awareness of the purposes to which it was being put. Yet the peculiar biochemistry of this plant species, its ability to concentrate sugars to such extraordinary levels, created economic incentives powerful enough to drive the forced migration of 12 million Africans across the Atlantic, making it perhaps the single most consequential plant in shaping the demographics of the modern world. While the Caribbean burned with sugar production, other botanical exchanges were proceeding more quietly, but with equally far-reaching effects. The potato, Solanum tuberosum, which originated in the highlands of Peru and Bolivia, arrived in Europe sometime in the late 16th century, but for decades it was regarded with deep suspicion. The potato belonged to the nightshade family, along with poisonous plants like belladonna and mandrake, and its above-ground parts are indeed toxic, containing solanine and other glycoalkaloids that can cause serious illness. Furthermore, the potato was not mentioned in the Bible, which for many religiously minded Europeans was reason enough to distrust it. It grew underground, in the realm associated with darkness and evil, and it produced tubers through a process that seemed vaguely unnatural, compared to the familiar sight of grain ripening in the sun. For a century after its introduction, the potato remained a curiosity, grown in botanical gardens and occasionally consumed during famines when nothing else was available. What finally changed European attitudes was a combination of demographic pressure, military necessity, and the advocacy of a few forward-thinking agriculturalists who recognized the potato's extraordinary nutritional properties. A single acre of potatoes could feed many more people than an acre of wheat, or rye, and the potato provided not just calories but significant amounts of vitamin C, potassium, and high-quality protein. Furthermore, potatoes could be grown on marginal land unsuitable for grain, could be left in the ground until needed rather than requiring storage, and were far less vulnerable to the kind of catastrophic weather events that could destroy a wheat harvest. In Prussia, Frederick the Great became an ardent promoter of potato cultivation, recognizing that a population fed on potatoes could support a larger army and sustain itself better during wartime. He distributed seed potatoes to reluctant peasants and supposedly ordered his troops to guard potato fields ostentatiously, calculating that people would want to steal and plant whatever seemed valuable enough to warrant military protection. Whether this story is apocryphal or not, the strategy worked in psychological terms. Within a generation, potatoes had become a staple crop across much of northern Europe. In Ireland, the transformation was even more complete. The cool, damp climate of Ireland was poorly suited to wheat, but ideal for potatoes, and the rural poor discovered that a family could survive on a small plot, planted entirely in potatoes, supplemented with a bit of milk or buttermilk. By the early 19th century, 1/3 of Ireland's population depended almost entirely on a single variety of potato, the Irish lumper, for their survival. This monoculture created a biological vulnerability of catastrophic proportions. In 1845, a water mould called Phytophthora infestans, which causes potato late blight, arrived in Ireland, probably on a ship carrying potatoes from the Americas. The organism spread with terrifying speed through the Irish potato crop, turning healthy plants into blackened, stinking masses of rotting vegetation within days. The spores could travel on the wind for miles, could survive in infected tubers left in the ground, and thrived in precisely the cool, wet conditions that characterized Irish weather. Over the next 7 years, repeated failures of the potato crop killed approximately 1 million people and drove another million to emigrate, reducing Ireland's population by a quarter. The British government's response to this catastrophe was shaped by laissez-faire economic ideology that deemed direct food aid to the starving as interference with market forces, a political philosophy that turned a natural disaster into a humanitarian tragedy. The famine, as it became known in Ireland, fundamentally altered the relationship between Ireland and Britain, fueled a diaspora that would reshape America and other destinations of Irish emigration, and demonstrated how the biology of a single plant species, when combined with social and political structures, could determine the fates of millions. But the potato blight also taught botanists and farmers crucial lessons about plant disease and the importance of genetic diversity. The Irish potato crop had been grown entirely from tubers, a form of asexual reproduction that meant every plant was essentially a clone of every other plant, genetically identical and therefore equally vulnerable to any pathogen that could successfully attack one plant. In the Andes, where potatoes had been cultivated for more than 7,000 years, farmers grew hundreds of different varieties, each adapted to specific elevations, soil types, and climate conditions, creating a buffer of genetic diversity that prevented any single disease from destroying the entire crop. This traditional knowledge, dismissed by European agricultural improvers as primitive and inefficient, actually represented a sophisticated understanding of risk management and ecological resilience that modern agriculture is only now beginning to fully appreciate. In the aftermath of the Irish famine, plant breeders began the long process of developing disease-resistant potato varieties and implementing crop rotation strategies, though these lessons would have to be relearned repeatedly as industrial agriculture expanded in the 20th century. Across the ocean in North America, a different plant was quietly preparing to reshape the economy and politics of an entire nation. Cotton, specifically Gossypium hirsutum, the species known as upland cotton, grew wild in Mexico and had been domesticated thousands of years ago in multiple locations across the tropics. The cotton plant produces fluffy fibers that surround its seeds, an evolutionary adaptation meant to aid in seed dispersal by wind and water. These fibers are almost pure cellulose, and when spun and woven, they create fabric that is comfortable, durable, and easy to clean. For most of human history, cotton cultivation had been labour-intensive but manageable, a crop grown in small quantities for local use. Two interconnected innovations transformed cotton into the foundation of the first global industrial economy: the mechanisation of textile production in England and the invention of the cotton gin in America. The gin, a simple machine developed by Eli Whitney in 1793, could separate cotton fibres from seeds 50 times faster than hand processing, suddenly making large-scale cotton production economically viable. This single invention breathed new life into the dying institution of slavery in the American South, created the textile mills of Manchester and Lancashire, and established trade patterns that connected Alabama cotton fields to Indian markets via British merchant ships. The cotton plant itself required specific growing conditions: a long frost-free period, moderate rainfall during the growing season, and dry conditions during harvest. The American South provided these conditions across vast territories, and cotton cultivation expanded with explosive speed, pushing westward into Mississippi, Alabama, Louisiana, and eventually Texas. Each acre of cotton required extensive labour: ploughing, planting, thinning, weeding, and then the back-breaking work of harvest, moving slowly down rows of waist-high plants in brutal heat, pulling the fluffy bolls from their sharp, rigid bracts that cut and scratched the hands of pickers. Enslaved people were forced to meet picking quotas that increased each year as overseers demanded ever greater productivity, and those who failed to meet their quotas faced savage punishment. The wealth generated by cotton was staggering. By 1860, cotton constituted nearly 60% of American exports, and the 4 million enslaved people forced to cultivate it represented more capital value than all American railroads and factories combined. This economic reality shaped American politics, leading to constitutional compromises that protected slavery, territorial expansion designed to create new slave states to maintain political balance, and ultimately to a civil war that killed more than 600,000 people. Yet the dominance of cotton also created economic vulnerability, as the Confederate States learned when Union naval blockades cut off cotton exports during the Civil War, and as Southern planters discovered repeatedly when cotton prices collapsed due to overproduction, or when insect pests devastated crops. The boll weevil, a beetle that feeds on cotton buds and flowers, entered Texas from Mexico in 1892 and spread eastward at a rate of about 40 to 160 miles per year, reaching the Atlantic coast by the 1920s. The weevil could complete multiple generations in a single growing season, and it reduced cotton yields by anywhere from 30 to 70% in infested areas. The insect's spread destroyed the economy of countless southern communities, accelerated the mechanization of cotton farming, and contributed to the great migration of African Americans from the rural South to northern cities. In Enterprise, Alabama, a town nearly ruined by the boll weevil, citizens erected a monument to the insect, recognizing that its destruction of cotton had forced economic diversification that ultimately made the town more prosperous. This paradoxical monument stands as a reminder that plants, and the insects that feed on them, can reshape human societies in ways both devastating and occasionally unexpectedly beneficial. While cotton was transforming American society, another fiber plant was shaping the maritime expansion of European empires. Cannabis sativa, cultivated for thousands of years across Eurasia for its strong stem fibers, became crucial to naval power in the age of sail. Hemp fiber, processed from the stalks of cannabis plants, was superior to other materials for making rope and canvas because it was strong, durable, and resistant to saltwater and rot. A single large warship required enormous quantities of rope, rigging, anchor cables, tackle for raising and lowering sails, lashings to secure cannon and cargo. The British Royal Navy's dominance of the seas depended on a steady supply of hemp, which they obtained initially from Russia and the Baltic region, creating a geopolitical vulnerability that concerned British strategists. The very word canvas derives from cannabis, indicating how central this plant was to the technology of sail. Colonial powers encouraged hemp cultivation in their territories, and in the American colonies farmers were sometimes legally required to grow it. Thomas Jefferson and George Washington both cultivated hemp at their Virginia plantations, and Jefferson even designed an improved hemp brake, a device for separating the valuable fibers from the plant's woody core. The process of transforming hemp plants into usable fiber was laborious, and required precise timing. The plants had to be harvested when the male flowers were mature, but before the seeds fully developed, then subjected to a process called retting, in which bundles of stalks were soaked in water or left in fields to allow bacteria and fungi to break down the pectin that bound the fibres to the woody stem. This process, if not carefully controlled, could either underret the hemp, making it difficult to process, or overret it, weakening the fibres. After retting, the hemp was dried, then beaten and scraped to remove the brittle woody material, leaving the long, strong fibres that could be spun into rope. The finest hemp came from Russia, particularly from regions where soil and climate conditions were optimal, and the disruption of hemp supplies during the Napoleonic Wars was one factor that motivated British efforts to develop alternative sources in India and other colonies. The transition from sail to steam power in the 19th century reduced the strategic importance of hemp, but the plant continued to be cultivated for rope, paper and textiles until synthetic fibres developed in the 20th century offered cheaper alternatives. In the forests of South America, a different plant was about to trigger an industrial revolution of its own kind. Hevea brasiliensis, the rubber tree, grew in the Amazon rainforest as a scattered canopy species. Individual trees spread out across vast areas rather than growing in dense stands. This distribution pattern was a defense against specialized pests and pathogens. If rubber trees grew close together, any disease that affected one tree could easily jump to its neighbors. The indigenous peoples of the Amazon had long known how to harvest the latex sap from rubber trees and use it to make waterproof containers, balls, and other objects. The tree produces latex as a defense mechanism. When the bark is wounded, the sticky white sap flows out and begins to coagulate, sealing the wound and trapping insects or other potential threats. The latex contains natural rubber, a polymer of isoprene, which gives it remarkable elastic properties. European scientists became aware of this material in the 18th century, but for decades rubber remained more of a curiosity than a useful industrial material because it became brittle in cold weather and sticky and malodorous in heat. In 1839, an American inventor named Charles Goodyear discovered, partly through accident and partly through years of experimentation, that heating rubber with sulfur transformed it into a stable material that maintained its elasticity across a wide range of temperatures. This process, which became known as vulcanization, turned rubber from a curiosity into an essential industrial material, used for everything from waterproof clothing to machine belts to, eventually, pneumatic tires. Demand for rubber exploded, and since Hevea brasiliensis grew naturally only in the Amazon basin, this created a rubber boom that brought enormous wealth to the Brazilian city of Manaus and to the rubber barons who controlled the trade. Manaus, a city hundreds of miles up the Amazon River, became fabulously wealthy. It built an opera house that rivaled those of European capitals, installed a tram system and electric lighting, while most of the world still relied on horses and gas lamps, and imported luxury goods from Paris and London. The rubber tappers who actually collected the latex, many of them indigenous people or poor mestizos, lived in conditions of brutal exploitation, often trapped in debt peonage to the rubber companies, forced to work in remote forest camps where disease and malnutrition were rampant. Brazil's rubber monopoly rested on the geographic distribution of a single plant species, but monopolies based on biological resources are inherently vulnerable to biopiracy and botanical smuggling. In 1876, an English adventurer named Henry Wickham collected 70,000 rubber tree seeds in Brazil and transported them to England, where they were germinated at the Royal Botanic Gardens at Kew. From these seeds, seedlings were distributed to British colonies in Asia, particularly Ceylon, Malaya, and Singapore. In Asia, freed from the natural pests and diseases of their native range, rubber trees could be grown in dense plantations that produced latex far more efficiently than the scattered wild trees of the Amazon. By 1914, Asian plantations were producing more rubber than Brazilian forests, and by the 1920s they had completely captured the global market, collapsing the Amazon rubber boom and plunging Manaus into economic depression from which it would not recover for generations. The magnificent opera house stood almost empty for decades, a monument to wealth built on a plant that no longer generated profit. This pattern, the transfer of economically valuable plants from their regions of origin to colonial plantations elsewhere, was repeated across the tropics, with coffee, tea, cinchona, and numerous other species, reshaping global trade and often devastating the economies of the plant's native regions. The cinchona tree, source of the antimalarial drug quinine, followed a similar trajectory. Native to the Andean forests of Ecuador, Peru and Bolivia, various species of cinchona had been used by indigenous peoples to treat fevers long before Europeans arrived. Spanish colonizers learned of the tree's medicinal properties, and Jesuit missionaries promoted its use in Europe, leading to its common name, Jesuit's bark. For centuries, Spain maintained strict control over cinchona bark exports, prohibiting the export of seeds or seedlings. The bark contains quinine and related alkaloids that can suppress the malaria parasite during its life cycle in human red blood cells, making it the only effective antimalarial treatment available until the mid-20th century. Control of quinine supplies therefore meant control over the ability to survive in tropical regions where malaria was endemic, which in turn meant control over colonial expansion. The British and Dutch desperately wanted to establish their own sources of cinchona, and through a combination of botanical espionage, diplomatic pressure, and outright smuggling, they eventually obtained seeds and plants. Dutch plantations in Java became the world's leading quinine producers, using carefully selected strains of cinchona that contained higher alkaloid concentrations than wild trees. When World War II cut off Allied access to Javanese quinine, the search for alternative antimalarials became a critical military priority, leading to the development of synthetic drugs like chloroquine and mefloquine. But even today, quinine remains in use for treating certain drug-resistant strains of malaria. The relationship between plants and human disease extends far beyond quinine's antimalarial properties. The opium poppy, Papaver somniferum, has been cultivated since Neolithic times for its psychoactive and analgesic properties. The plant produces morphine and related alkaloids in specialized cells called lattice-ciphers, which run throughout the plant but are most concentrated in the seed capsule. When the unripe seed capsule is scored with a knife, latex containing these alkaloids seeps out and can be collected and dried to produce raw opium. This process has been refined over millennia, particularly in regions of Southwest Asia and later in China, India and Southeast Asia. Morphine, the primary active compound in opium, is arguably the most effective painkiller ever discovered, capable of relieving even the most severe pain by binding to specific receptors in the brain and spinal cord. Yet this same mechanism creates profound addiction, as the brain adapts to the constant presence of the drug and develops dependence. The trade in opium became a flashpoint in international relations in the 19th century, particularly in the opium wars between Britain and China, conflicts that were ultimately about Britain's desire to use opium exports to balance the massive trade deficit created by British imports of Chinese tea. Tea itself, Camellia sinensis, was another plant whose cultivation and trade shaped empires. Native to the borderlands between Burma, China and India, tea had been consumed in China for thousands of years, but it only became known in Europe in the 17th century. The Chinese tea industry was extraordinarily sophisticated, with different regions specializing in different varieties and processing methods. Green tea, which is steamed or pan-fired quickly after harvest to prevent oxidation. Black tea, which is fully oxidized before drying. Oolong, which is partially oxidized, and numerous regional specialties. The process of making tea requires careful attention to detail at every stage. The timing of the harvest, with the youngest, most tender leaves producing the finest tea. The specific methods of withering, rolling, oxidizing and drying, and the precise control of temperature and humidity throughout. Chinese tea producers guarded their methods carefully, and for centuries China maintained a near-total monopoly on tea production. Britain, which had developed an intense cultural attachment to tea drinking in the 18th and 19th centuries, faced a massive trade imbalance as silver flowed to China in payment for tea, while China bought relatively few British goods in return. The British response was multifaceted and ruthless. They promoted opium cultivation in India and smuggled it into China to create demand for a British-controlled product, and they engaged in what can only be called industrial espionage to steal tea plants and production knowledge from China. In 1848, the British East India Company sent a Scottish botanist named Robert Fortune into the interior of China, disguised in Chinese dress and travelling with a shaved head and queue to collect tea plants and learn the secrets of tea processing. Fortune succeeded in obtaining thousands of tea plants, and in recruiting Chinese tea workers who could train Indian workers in proper tea cultivation and processing techniques. These plants were established in the Himalayan foothills of Assam and Darjeeling, regions whose climate and elevation could support tea cultivation. Within decades, British India became a major tea producer, breaking China's monopoly and ensuring that Britain's tea supplies would flow from within its own empire. The social and economic effects of this shift were profound. Tea plantations in India and later in Ceylon became major employers and generators of export revenue, though the working conditions on these plantations were often appalling, with workers trapped in debt bondage and subjected to harsh discipline. The global movement of plants accelerated dramatically in the 19th and 20th centuries, creating ecosystems that would have been unrecognizable to anyone living before the age of exploration. Eucalyptus trees from Australia were planted across the Mediterranean, California, South America, and Africa, valued for their rapid growth and ability to thrive in poor soils, but becoming in some cases invasive pests that consumed scarce water and fueled intense wildfires. The American prickly pear cactus, introduced to Australia as an ornamental and for agricultural purposes, spread across millions of acres, creating impenetrable thickets that made land unusable for farming or grazing, until biological control agents, insects that feed on prickly pear, were introduced in the 1920s. Water hyacinth, a beautiful aquatic plant from the Amazon, was distributed around the world as an ornamental for ponds and water features, then escaped into rivers and lakes where it formed dense mats that blocked waterways, disrupted fisheries, clogged hydroelectric dams, and created breeding grounds for disease-carrying mosquitoes. The kudzu vine, introduced from Japan to the United States for erosion control and forage, grew with such abandon in the warm, moist climate of the American South that it came to symbolize the unintended consequences of botanical introduction, covering abandoned buildings, telephone poles, and entire forests in a smothering green blanket. These biological invasions were not merely aesthetic or agricultural problems, but represented fundamental disruptions to ecological relationships that had evolved over millions of years. Native plants had evolved in the context of specific pollinators, herbivores, soil microorganisms, and competitors, creating intricate webs of interdependence. When a plant species moved to a new environment, it often left behind the insects, diseases, and other organisms that had controlled its population in its native range, allowing it to grow without the biological constraints that had limited its spread in its homeland. The emerald ash borer, an Asian beetle accidentally introduced to North America, has killed hundreds of millions of ash trees because American ash species, having evolved without this pest, have no defenses against it. The chestnut blight, a fungal disease from Asia, eliminated the American chestnut as a dominant forest tree in the early 20th century, fundamentally altering the ecology of eastern North American forests and destroying a resource that had provided food and timber to both wildlife and human populations. Yet the same processes of botanical exchange that created invasive species and ecological disruptions also saved millions of lives through the global dispersal of food crops. The sweet potato, native to tropical America, was carried by Spanish and Portuguese traders to the Philippines and from there to China, where it became a crucial crop in regions with poor soil or unreliable rainfall. By the 18th century, the sweet potato allowed Chinese farmers to cultivate marginal lands that could not support rice or wheat, contributing to a population boom that brought China's population from around 150 million in 1700 to over 400 million by 1850. Maize, originally domesticated in Mexico from a wild grass called teosinte, in a transformation so complete that botanists argued for decades about its origins, bred around the globe after Columbus, and became a staple food in regions as diverse as sub-Saharan Africa, southern Europe, and the American South. The process by which teosinte, a plant with small, hard-shelled seeds arranged in a simple spike, was transformed over generations of selection by Mesoamerican farmers into modern maize, with its large ears containing hundreds of plump, starchy kernels held on a rigid cob, represents one of humanity's most remarkable achievements in plant breeding, accomplished without any understanding of genetics or heredity, purely through careful observation and selection. The domestication of plants, in fact, represents a co-evolutionary relationship in which both humans and plants were transformed. Wild wheat and barley grew in the Fertile Crescent as scattered stands of grain that shattered readily when ripe, scattering their seeds to ensure reproduction. Farmers gathering these grains would naturally collect more from plants whose seed heads held together longer, inadvertently selecting for a mutation that prevented seed dispersal. Over generations, this selection pressure transformed wild grasses into domestic grains that depended entirely on humans for their reproduction, since their seeds remained trapped in non-shattering heads unless harvested. Humans, in turn, were transformed by their relationship with these grains. Farming communities became sedentary, developing permanent settlements that grew into villages, towns, and eventually cities. They created new tools for ploughing, harvesting, and processing grain. They developed systems of storage and redistribution that led to social hierarchies and specialized occupations. And they altered their own biology, as populations that relied heavily on grain developed genetic adaptations for digesting starches and for surviving on diets that, while providing abundant calories, were nutritionally less diverse than the hunter-gatherer diets they replaced. The archaeological record reveals the physical costs of this transition. Early farming populations show increased rates of dental disease, anemia, infectious diseases, and skeletal markers of nutritional stress compared to earlier hunter-gatherers. The concentration of populations in permanent settlements created ideal conditions for the transmission of infectious diseases, many of which were zoonotic diseases that jumped to humans from domesticated animals. The heavy reliance on a few staple crops meant that crop failures could lead to devastating famines. Yet the sheer caloric productivity of agriculture allowed human populations to grow far beyond what hunting and gathering could support, creating demographic momentum that made any return to previous subsistence strategies impossible. The plants that humans domesticated, wheat, rice, maize, potatoes, cassava, soybeans, and others, now occupy vast areas of the Earth's surface, areas that are managed, irrigated, fertilized, and defended against competing plants and hungry insects with an intensity that would astonish farmers from just a century ago. Modern industrial agriculture has achieved productivity levels that seem miraculous when measured against historical norms. Average corn yields in the United States have increased from about 25 bushels per acre in 900 to over 170 bushels per acre today, a nearly seven-fold increase. This transformation was accomplished through hybrid seed development, synthetic fertilizers, pesticides, irrigation, and mechanization, each of which carries its own costs and consequences. The Haber-Bosch process, developed in the early 20th century to synthesize ammonia from atmospheric nitrogen and hydrogen, made possible the production of synthetic nitrogen fertilizers that allowed crop yields to soar, but it also created a dependence on fossil fuels both for the energy required to run the process and for the hydrogen feedstock derived from natural gas. Nitrogen fertilizers applied to fields but not fully taken up by crops wash into rivers and eventually into oceans, where they fuel algal blooms that, when they die and decompose, create dead zones devoid of oxygen and therefore of most aquatic life. The Gulf of Mexico dead zone, which appears each summer at the mouth of the Mississippi River, can cover an area the size of New Jersey, a direct consequence of nitrogen runoff from cornfields in the Midwest. The pesticides that protect crops from insects, weeds, and diseases have become increasingly sophisticated, targeting specific biochemical pathways in pest organisms, but they have also generated resistance in the very organisms they were meant to control. Insects and weeds evolve rapidly, and populations subjected to intense selection pressure from pesticides can develop resistance mechanisms within just a few generations. Glyphosate, the world's most widely used herbicide, was considered nearly miraculous when it was introduced in the 1970s, because it killed a broad spectrum of weeds while being relatively low in toxicity to mammals and decomposing quickly in soil. The development of glyphosate-resistant crops through genetic engineering in the 1990s allowed farmers to spray entire fields without harming their crops, leading to massive increases in glyphosate use. Inevitably, weeds began to evolve resistance. Palmer amaranth, a weed that infests cotton and soybean fields, has developed resistance to glyphosate and multiple other herbicides, and it grows so aggressively that it can reduce crop yields by 50% or more, and grow so dense and tough that it can damage harvesting equipment. The plant at the center of these modern agricultural dramas, whether corn or soybeans or cotton, is the product of breeding programs that have radically altered the genetic makeup of crops through selection for traits like high yield, uniform ripening, disease resistance, and responsiveness to fertilizers. Modern hybrid corn, for instance, bears little resemblance to the corn grown by American farmers in the 19th century, let alone to the diverse landraces cultivated by indigenous farmers in Mexico and Central America. This genetic narrowing has created crops of extraordinary productivity under optimal conditions, but also of potential vulnerability to new pests, diseases, or climate conditions. The 1970 corn blight epidemic in the United States, caused by a fungus that specifically attacked a type of male sterile cytoplasm that was present in about 85% of American corn hybrids, destroyed about 15% of the national corn crop and demonstrated the risks of genetic uniformity. The epidemic was brought under control by switching to different corn varieties, but it highlighted the importance of maintaining genetic diversity as insurance against future threats. Meanwhile, in research laboratories and botanical gardens, scientists race to collect and preserve the genetic diversity of crop wild relatives before it disappears. The wild ancestors and close relatives of domestic crops contain genes that have been lost from cultivated varieties, genes for drought tolerance, pest resistance, salt tolerance, and other traits that may become crucial as climate change alters growing conditions around the world. Centers of crop diversity, places like the highlands of Ethiopia for wheat and barley, the Andes for potatoes, Mesoamerica for maize and beans, are under intense pressure from habitat loss, climate change, and the replacement of traditional crop varieties with modern commercial varieties. Seed banks, such as the Svalbard Global Seed Vault buried in the permafrost of a Norwegian island, store millions of seed samples as backup copies of agricultural biodiversity, insurance against catastrophic loss. Yet seed banking is not a perfect solution. Some seeds do not remain viable long in storage. The stored seeds are not evolving in response to changing conditions, and the traditional knowledge about how to grow and use rare varieties is being lost as rapidly as the seeds themselves. The acceleration of plant extinction parallels the loss of crop diversity. Current extinction rates are estimated to be 100 to 1,000 times higher than background rates, and plants are disappearing before they have even been scientifically described. The Amazon rainforest alone may contain tens of thousands of plant species unknown to science, and these forests are being cleared for cattle ranching and soybean production at rates that vary from year to year, but have at times exceeded 10,000 square kilometers annually. Each plant species represents millions of years of evolution, a unique combination of genes and biochemical pathways that might produce compounds useful for medicine, agriculture, or industry. The rosy periwinkle, a small plant from Madagascar, produces compounds that have proven effective in treating certain cancers, but it was nearly lost to extinction before its medical value was recognized. Madagascar has lost more than 90% of its original forest cover, and with it countless species that existed nowhere else on Earth. Yet even as extinction proceeds, plants continue to shape the atmosphere and climate of the entire planet through processes operating at timescales that dwarf human history. Photosynthesis, the process by which plants use light energy to convert carbon dioxide and water into sugars and oxygen, has maintained Earth's atmospheric oxygen at roughly 21% for hundreds of millions of years, creating the conditions that made possible the evolution of large active animals, including humans. Every breath a human takes contains oxygen produced by photosynthesis, much of it from phytoplankton in the oceans, but a substantial fraction from land plants as well. The Amazon rainforest alone produces an estimated 6 to 9% of the world's oxygen, though it also consumes most of this through respiration and decomposition, making its net contribution to atmospheric oxygen minimal. However, the forest's role in the carbon cycle is profound. The trees and soils of the Amazon store somewhere between 150 and 200 billion tons of carbon that would otherwise be in the atmosphere as carbon dioxide, intensifying the greenhouse effect and accelerating climate change. As global temperatures rise and precipitation patterns shift, plants are responding in ways both predictable and surprising. The growing season in temperate regions has lengthened as spring arrives earlier and fall extends later. Tree lines are moving up slope on mountains and poleward in latitude, and plant species ranges are shifting as climate zones migrate. Some plants are responding rapidly, tracking changing conditions through dispersal and establishment in newly suitable areas, But many others, particularly long-lived trees, are responding more slowly, creating mismatches between climate and vegetation that may persist for decades or centuries. The boreal forests that encircle the Arctic, composed primarily of spruces, firs and larches, are experiencing warming at twice the global average rate, leading to increased stress from drought, fire and insect outbreaks. The spruce bark beetle, favoured by warmer temperatures, has killed millions of acres of spruce forest in Alaska and western Canada, transforming landscapes that had been stable for thousands of years. In the oceans, warming water temperatures and increasing carbon dioxide levels are affecting not just fish and corals, but also the phytoplankton that form the base of marine food webs and produce much of Earth's oxygen. These microscopic plants and plant-like organisms are responding to ocean acidification, changes in nutrient availability, and shifting current patterns in complex ways that scientists are only beginning to understand. Some species of phytoplankton may benefit from higher carbon dioxide levels, at least initially, but changing ocean chemistry could disrupt the delicate calcium carbonate structures of coccolithophores and other calcifying plankton, with cascading effects throughout marine ecosystems. On land, plants face not only changing climate, but also changing atmospheric composition directly. The concentration of carbon dioxide in the atmosphere has risen from about 280 parts per million before the Industrial Revolution to over 420 parts per million today, a level not seen on Earth for at least 3 million years. This increase affects plants directly because carbon dioxide is a raw material for photosynthesis, and some plants, particularly those using the C3 photosynthetic pathway, respond to elevated carbon dioxide with increased growth, at least under conditions where nutrients and water are not limiting. However, this carbon dioxide fertilization effect is not uniformly beneficial. Plants grown under high carbon dioxide often produce tissues with lower nutrient content and different chemical composition, which can affect everything from insect herbivory to human nutrition. The wheat, rice and soybeans produced under future high carbon dioxide conditions may contain lower concentrations of protein, iron and zinc, potentially exacerbating malnutrition in populations that depend heavily on these crops. Meanwhile, weeds, many of which are C4 plants less responsive to carbon dioxide fertilization, but more tolerant of heat and drought, may gain competitive advantages over crops, shifting the balance in the ancient struggle between farmers and the plants they have not chosen to cultivate. Poison ivy, a C3 plant that responds vigorously to elevated carbon dioxide, grows faster and produces more potent urushiol, the compound that causes its characteristic rash under high carbon dioxide conditions, suggesting a future in which this already unpleasant plant becomes both more abundant and more toxic. These ongoing transformations underscore a reality that has been constant throughout human history. Plants are not passive resources but active agents in their own right, responding to environmental conditions, competing for light and nutrients, defending themselves against herbivores and pathogens, and reproducing according to strategies honed over millions of years of evolution. The relationship between humans and plants is not one of simple domination, but of mutual entanglement, a relationship in which human societies have been shaped by the particular properties of the plants they cultivate and consume, just as surely as humans have shaped those plants through selection and breeding. The coffee that fueled the Enlightenment, the potatoes that sustained population growth in Northern Europe, the cotton that drove American slavery and British industrialization, the rubber that enabled the automobile age, the wheat that fed empires and whose failure sparked revolutions. These plants were never merely passive materials, but participants in historical processes that neither the plants nor the humans could fully control or predict. As human population approaches 8 billion, and as demands for food, fiber, fuel, and living space intensify, the relationship between humanity and the plant kingdom enters a phase of unprecedented stress and transformation. A transformation whose outcome will determine not just human welfare, but the structure of life on Earth for millennia to come. A future being written in the genetics of seeds stored in frozen vaults, in the composition of the atmosphere itself, in the expanding and contracting ranges of species responding to climate change, and in the agricultural fields where farmers continue the ancient work of coaxing from plants the sustenance that makes human civilization possible, while the plants themselves continue their even more ancient work of capturing sunlight and transforming it into the complex molecules of life, a collaboration and conflict between kingdoms that has shaped the history of this planet and will shape its future in ways both terrible and wondrous beyond our current capacity to imagine or control.